All right, so today we're going to talk about details of meiosis. And the first thing here is that we have interphase. An interphase is going to be the same as it is for any type of cell division, so if we're doing mitosis or meiosis. So we'll still have our G1, our S, and our G2. All of those things are going to happen. For pictures, you'll still see the normal cell, the normal nucleus, which has chromatin on the inside, and then your centrosomes. And then when we're going into meiosis, okay, again, for meiosis 1, we'll start out with prophase 1. What you'll see is just one cell. The nuclear membrane is going to start to be breaking down. Your spindles are forming and your centrosomes are moving away from each other, with the spindles forming in between. And then the big, huge difference here is that our homologous chromosomes are pairing up and forming tetrads, and you'll also have crossing over taking place. So crossing over, homologous chromosomes pairing up, forming tetrads, those are big indicators that we're talking about prophase 1. Then we move right into metaphase 1, again, one cell. And the thing that you'll notice here then is that we have homologous chromosomes lining up in the middle. So notice we have like one X is sort of next to the other chromosome that looks like an X, and so one's like these guys are all like on the north side and these guys are all on the south side of the equator, and then the spindles will come down and attach. So same metaphase in the middle, but now we have homologous chromosomes lined up in the middle. In anaphase, anaphase 1, we're still moving chromosomes apart from each other, except this time they are duplicated chromosomes, so the homologous chromosomes are separating and moving away from each other. So homologous chromosomes are separating, that's key. For telophase 1, you'll notice it looks the same. We have the cleavage furrow in animals, the cell plate will form in plants, so cytokinesis is starting to happen during telophase. Again, reverse of prophase, so the nuclear membrane might be coming back for a bit. Depending on how long it'll take place, maybe these guys will unwind, maybe they'll stay as chromosomes. But notice they're still duplicated chromosomes, but the number of chromosomes that are in each of the cells is cut in half. So at this point, these are haploid cells, because the chromosome number is cut in half, but they are duplicated chromosomes, and so that's the big difference. Okay, so telophase 1 and cytokinesis. Then we go right into prophase 2, which will then consist of all these stages, and there's no interphase in between. So we go right from cytokinesis, telophase 1 and cytokinesis, into prophase 2. There's no interphase in between. Notice, for these pictures here, you'll see 2 cells, and so that's going to be a good indicator that we're looking at something with a 2 behind it. Again, all of these cells are going to be haploid. For prophase 2, you'll see the nuclear membrane is, breaking down, so it looks kind of like a normal prophase, like you would see in mitosis. It's just that we have 2 cells going on, and then there'll be half the number of chromosomes. In metaphase, we have the chromosomes lined up in the middle. Okay, and again, 2 different cells with chromosomes lined up in the middle for metaphase 2. In anaphase 2, again, 2 cells where now the sister chromatids are separating. So again, this looks a lot like mitosis. It's just that we have 2 cells going on, and both of those cells are haploid. But the sister chromatids are moving. They're now individual chromosomes, and they're moving towards the poles for anaphase 2. And then for telophase 2, again, the nuclear membrane is going to be coming back. The chromosomes are going to be unwinding into chromatin. We'll start to have cytokinesis. So we'll start pinching in animals, or we'll be forming cell plates in plants. Sometimes they'll show like 2 cells that are looking like little peanuts, or the pinched in cells. sometimes you'll actually see 4 different cells. And so if you see 4 cells, huge indicator that we're looking at telophase 2. Or if you see 2 cells that are going through cytokinesis with the pinches, again, another indicator that it's telophase 2. All right, so I hope that helps. If you have any questions, go back and re-listen or re-watch. Please ask questions in class. Hope you have a great day. Make good choices, and Jesus loves you.